My name is Panyu Chen, and I'm a cancer biologist by training. I studied cancer biology for both PhD and postdoc. So together, I've been working in the field of cancer research for 15 years now, with the goal of providing improved treatments for cancer patients. Currently, I'm leading discovery and translational activities as the Associate Director of Translational Medicine at Opna Bio, which is a clinical stage biotech company based in the San Francisco Bay Area. So Opna Bio is focused on developing therapeutics for heme malignancies, including multiple myeloma and myelofibrosis. We have 2 clinical programs, and the project that I'm presenting this Sunday is actually on a novel discovery program that we have. It was motivated by data from our clinical compound 6602, which is a small molecule oral inhibitor of EP300 and CBP that is currently being evaluated in the phase 1 clinical study in patients with relapsed and refractory multiple myeloma. The indication we're addressing is multiple myeloma, which is a type of blood cancer that arises from abnormal proliferation of plasma cells. And in terms of prevalence, worldwide, every year there's estimated to be 160,000 new cases of multiple myeloma, making it the second most common heme malignancies. With the degrader program, we would like to address some limitations with the current standard of care therapies for multiple myeloma, including the issue of resistance, as well as being able to make it into an oral drug. I'll start by giving some background on how our degraders work. So IMiDs, or immunomodulatory drugs, such as lenalidomide and pomalidomide, are currently a standard of care for multiple myeloma treatment. And the way they work is by degrading Ikaros and Aiolos, which are important targets for multiple myeloma treatment. And even though IMiD drugs are highly effective, resistance often occurs in patients, and one possible way to overcome such resistance is through EP300 and CBP inhibition, which our compound OPN6602 can achieve. So we had this idea of combining the activities of IMiD drugs with 6602 into a single drug to achieve single agent anti-myeloma activity. By combining activities of IMiDs and 6602, we can simultaneously cause elimination of cancer-promoting proteins, including Ikaros, Aiolos, EP300, and CBP, all in the same cancer cell. You can think of the catalytic activity somewhat like having a drug that acts like a serial killer. So once the drug gets into cancer cell, it can degrade each target sequentially, achieving a very high potency. So being able to dose this single drug, we can kill off cancer cells and have better control of the safety profiles as compared to if you had to dose multiple drugs. Right now, we already have achieved pretty high potency with our degraders, so we are very focused on improving the overall pharmacological properties, including oral bioavailability and tolerability. So this is where our medicinal chemistry efforts are going right now, and the goal is to have a lead candidate selected by mid-2026 and to file an IND in 2027. Our first goal is to create an oral drug, which means that the patients, instead of having to be treated in the hospital, can now bring home a bottle of pills and just take them at home. So by doing so, we hope to make the treatment experience more manageable and more convenient for patients. Secondly, we would like to achieve better durability and improve efficacy with our compound.